Yeah, let me start by welcoming you all. It's a pleasure to have you all online here for this workshop. It's really unfortunate that we can't do this live in town as was originally planned, but we've put together a little bit of a package, and we hope that it will be as lively as possible. So after I give a short theoretical or overview or just a talk through of activities and our expertise and what ecophysiology is all about, we'll move to the glass house and have some of the demonstration that we were intending to do live. We'll do them live, but we won't be there physically, but we hope to make it as interactive as possible. So I will start by sharing the screen, and bear with me. So I hope you'll soon see the start of the presentation, the title slide coming up.
Yeah, we can see it, Eric.
Excellent. Thank you very much for confirming that. So what we're trying to do today is to talk about the role of ecophysiology in mine site restoration. And of course, we've been part of the Center for Mine Site Restoration over the past few years and had lots of activity going on, building on previous work in the last 20 years or so, where we've pioneered the use of ecophysiology in many different mining situations, always associated with restoration or with impact assessment. So I'm presenting this on behalf of the team that I'll introduce shortly. I am in the School of Biological Science at the University of Western Australia, and this is a talk about ecophysiology. But over the past few years, the real work has been done by this team here. On the top, we have Wei-san, Jaume, Siwan, PhD students enrolled at UWA and active in the area of ecophysiology. Christine enrolled at Curtin, and Alina at UWA as master's students. And we've had postdocs as well involved in the ecophysiology team. Originally, it was John Young for over a year, and then Justin after a break for another year, and Fiamma is finalizing everything and leading the team now. And Hong Tao is assisting with all sorts of activities, and that has happened for last year now as well. So we'll meet some of them in the greenhouse shortly, and I want to thank them all at this point for all their valuable contributions. So when we talk about plant physiology, what are we really talking about? So the aim of plant physiology is to understand plant function, how they respond to the environment, and I say above and below ground, because below ground is where a lot of things happen in the environments that we are dealing with here, and how that allows them to thrive in their habitat, how they are adapted to it, and how they are resilient in that environment. So we focus on things like how they capture resources, important things like light, water, nutrients, and also CO2. So we look at things, processes like growth, photosynthesis, transpiration, the transport of these nutrients, and so on, and how plants respond to stress. And there's lots of stresses in our environments: temperature, radiation, drought, all sorts of challenges in the soil, environment, physical, chemical, and biological. And we look at the whole plant life cycle. So we don't just look at mature plants. In fact, a lot of the work that we do is on that early stage. There is, of course, a seed program as well in the CMSR, and seed biologists are really specialized at that usually important stage of getting plants germinated. But as soon as plants are germinated, ecophysiologists look at how they establish and survive. So in a practical sense, when you look at a given restoration site, we we can answer any of this sort of questions of, did this plant die of drought stress? Why is this plant growing faster than the other? Can this shrub fix nitrogen, and does it actually do it? Why do we get weeds? Why do they grow better than natives? Why do they compete more? In problematic situations, what are the issues? What are the challenges that plants deal with, and how can we overcome them? Is it important or not that there's so many rocks in the substrate? And ultimately, we think that those sort of questions and answers are very useful in mine site restoration. So it helps us make decisions about site preparation, about how and when to sow or plant, and what sort of interventions might be useful after things start going in a direction that is not fully desirable. We can even help making decisions about species selection. There may be sites where certain species we can predict that they just will not be successful, knowing their physiology, their functions. And we can diagnose issues based on plants growing in the field that don't seem to do so well. We can diagnose what's wrong with them. Is it nutrients? Is it water? Is it something else? Many mine sites also deal with situations where they have to clear some native vegetation that has rare species and common species. The rare species sometimes have to be moved. Populations need to be established in alternative sites, and we can help identify that sort of sites. After establishing a new restored site, there has to be a lot of monitoring, and the traditional monitoring is based on vegetation surveys with relatively little attention for plant condition. We think that plant conditioning monitoring would really help in diagnosing issues and have a more solid understanding of how well the restoration vegetation is performing. So altogether, we think we can help by predicting how well the outcomes are being achieved with the restoration trials. So concretely for this workshop today, what we think we're able to offer today is to give you some introduction to some basic principles which will set the scene for understanding the things that we measure and for the demonstrations that follow. And in these principles, I will focus on water, a little bit on stress in general, but it'll be a little bit of a bias for water, partly because that's what we're specialized in, that's what we've done most of our work on, but also obviously because it's one of the main issues that plants have to deal with in almost all of the Western Australian environments that we've worked in. So I'll then, at the end of this presentation, give some introduction to what the key methods are, flowing into a part that looks at proxy measures, easier ways of doing things than the often complicated ways that we use. And then we will dash down to the glass house where the rest of the team is preparing to demonstrate methods, and that's what then will conclude the session with a chance to ask your questions, get some answers from us, and any further discussion as required. I haven't mentioned that it will be a little bit difficult to answer questions during this presentation, in this first half hour or so, but I'm of course happy to answer those questions at the end or in the course of the workshop. So one thing I think that will be very obvious to any of you listening is that plants in Western Australian environments face many challenging conditions. And that's true for native ecosystems, but it's even more true for restoration conditions. In restoration environments, substrates are usually different to the natural condition. They may be unstable, they're different in terms of their geological materials, their textures, their rock content, certainly biologically, because topsoil availability is usually insufficient. And if if there is topsoil available, often its quality is deteriorated because it has to be stockpiled. The climate is an issue in native systems. Plants are adapted to it, but the climate is harsher in environments, in restored environments, because they're more exposed, they're altered, they're not stable substrate, there's no adult vegetation to offer protection and so on. So we look at those stresses, and in the next few slides I'll give a few three sort of the main groups of attention, starting with soils. And there's lots of issues in soils. There can be nutritional issues, there can be salinity, acidity, sodicity, there can be heavy metals, toxic metals. There can be a lack of carbon, and of course the biological side, all the microbes, is a big unknown, increasingly well known, but still pioneering stage. So for many of these things we can do diagnostic tests, so we can send off, and often we do, samples to labs to get tests, but their interpretation is often full of difficulties, and their correction is sometimes possible, like in the case of fertilizers, to correct nutrient issues. We can, for example, inoculate to make sure that there's rhizobia for nitrogen fixing plants. We can add sources of organic matter, but how much, what form, when, etc., is not always obvious. I think it's important to be realistic about substrates. If we are starting restoration on substrates that are really different from the original substrates, it will be very hard to expect that we can re-establish all the native plants at the very same site in similar abundances. The reality of these natural sites is that they're invariably formed on long formed soils. Over geological time, there has been evolutions of interactions between species, plants and microbes. The soil formation, including the impoverishment, the low levels of nutrients that they've gotten used to, has taken a long time. New substrates are not necessarily the same. Now, a few more words about life in soils. There is a lot of life in soils, certainly only partly explored, partly known, but increasingly well known due to novel approaches like genomic approaches, in DNA fingerprinting, microbial biome assessments, which are quite easy nowadays, but still have lots of issues in interpretation associated with them. But there's a lot of growth in this area, which I won't be talking about. You might hear about that in other workshops. So just to summarize a little bit what our expertise is and what sort of things are going on in this space. So we, in this group here at UWI, we have a lot of experience with plant adaptations regarding nutrient availability, particularly plants that are favorable in terms of making available nitrogen, phosphorus, and micronutrients to themselves and to other plants, so which can be engineering the site, making it more suitable for the rest of the vegetation. And that's a form of facilitation. Facilitation is the opposite of competition. Facilitation is making sites better suited to other species. And there's an interesting aspect of complementarity there between different species doing different things. I've talked about the microbiome already. We started some work, and Wei-San and John Young have been instrumental in that. We look at the microbiome, also followed by Justin later on in collaborations with the Curtin crew on what difference they might make to growth and stress tolerance. Temperature is another big thing, and one certainty about the current climate change events is that we are in a warming world. We already are a warm continent. Many of our study sites are very warm, and heat waves will be a major concern. Rehab sites very exposed, and the heat stress is combined with drought stress, often fatal. So we definitely need to look into heat itself, as well as interaction with drought. The difficulty in studying heat is often that a short spell of heat can do a lot of damage, and you are not often on site to witness that and to measure that. So it's not always easy to recognize it. And there's also cumulative heat stress damage that accumulates over time in over summers, for example. So it's not easy to diagnose, and it's not easy to remediate either, because there's very little you can do about the climatic events of heat stress. One thing that has been offered, one potential option that has been offered to deal with this changing climate is to use species and provenances that are more suited to those heat stress events, more tolerant to that. And of course, many of our species have a reasonably wide distribution. So you could look at choosing the provenances for those species that have that tolerance. Still partly controversial, but things that we definitely have started looking at, and there's currently an AOC linkage funded project going on for this one coastal plain, looking at some of the main species. So some of the research going on, I just mentioned one example, but we have been doing experiments where we are testing how heat stress and water stress interact. That's something that is really worth doing, because there's not a lot of that yet around. We're looking at stress indicators very broadly to make that suitable and to diagnose and assess the extent of stress. And we'll be assessing thermal tolerances of species like the provenances that I've mentioned before. Also worth mentioning that heat and temperature in general is something that can be remotely sensed quite well. So it's something that we can document quite well and have been looking into, as you will see soon. Now, finally, a large area of research is about water. We call that water relations of plants, and it really, in our environments, relates to the reality that there's large parts of the year that there's a lack of water, or almost the entire year. Australia is very dry, and on top of that, some parts are even getting drier. So we have been looking at water availability, this term that we often use at rehab sites. They often are hydrologically and climatically a little bit different from the natural sites. So water availability is not necessarily the same at the rehab sites as the natural sites. And as I've said, for the heat, combination of water stress and heat stress is often fatal. Now, when we look at water issues, we have to realize that dehydration of plants can be due to both the soil side, that there's too little water there, or that plants cannot access it, or that water loss is too high, or both of these. We can not do much about the climate. What is done in horticultural agriculture situations and in your garden is that you can irrigate if it's get too dry. That's usually not an option in mine site environment, so we need to look at tolerance. So because lots of these issues are actually below ground, we know from our research and from theory that substrates can make a big difference. The depth of substrates, the rootable depth, the textures, and so on. And again, for this also, we can look at climate-adjusted provenancing. So we really know a lot about drought stress in the Australian environment, and we've done a lot of experiments on control conditions and in the field. And we're looking at issues like texture, rock content, and impedance for root growth, which means that roots can have less access to volumes of soil, which means less water. In all of this, we use a range of techniques, and some of we will demonstrate for you, others we cannot, because they're really field techniques, like trees that flow things. But many of those are fairly costly, are difficult to replicate, difficult to do enough measurements over a short time or over long times. So we're looking at remote sensing for this as well, and we'll show you some potentials there in the demonstration. So in the following slides, I'll go into a little bit more detail about the physiology of water availability, because that's what most of our demonstrations relate to, and that's what our major area of research is about. So the big question in much of this is how much water is really available to those plants. And this issue of water availability, this concept, is not just about rainfall. It's about the water balance of an ecosystem, and it's about how much moisture the roots have access to, and how much it needs to replace the water it loses through transpiration. So it's not rainfall that matters, it's really the amount of water that ends up in soil. And on the right hand side you see this diagram which shows the major hydrological fluxes that determine what ends up in the soil. And plants have a major role in that, because they are the conduit of removing water from the soil into the air again. So this is a general image, and in some systems you have zero drainage, like some of the most arid systems. In other systems you might have lateral run on or run off. This is just a simplification, of course. But it's really important to look into these things, and we therefore do a lot of ecohydrology along with the ecophysiology. So we don't just look at the plants, but at the system and the hydrological fluxes in collaboration with hydrologists and soil scientists. And one illustration of why it's so important to understand that is this aerial picture from the Great Sandy Desert, where you have trees in the desert. They're decent sized trees, and you find them associated with the highest points in the landscape, often opposite to what you find in higher rainfall places where you might find lower vegetation on the dunes. Now, the understanding of that is in understanding the infiltration and the storage of water, because those trees are on the dunes because that's where they have access to most of the water. On the swells, you get the same rainfall, but there's no capacity to store that water, and there's no root access to great depth because there's a hard pan underneath it. So look at the plants in a little bit more details then. Plants, of course, hold a lot of water in the tissues. Almost any plant will have more than 50% water in its tissues. Some will have more than 90% water in their tissues. But apart from that, they use up to 100 times more water through water uptake than what's actually held in the tissues. So that is an enormous amount of water that flows through the plant just without being used to build tissues. And that comparison also shows that as soon as there's an imbalance between transpiration and uptake, plants dehydrate really very quickly because of that fast outflow. So what can plants do to avoid that dehydration is to really regulate the transpiration very tightly. And there's several mechanisms that they can employ here. They can manipulate their leaf area, grow more or less leaves, drop leaves when it's too stressful. They can grow a lot of roots deep and also extensive wherever the water is. And the tissues of leaves and so on can have an anatomy that's particularly suited to tolerate dehydration. So the mechanics of it all, for the mechanics, we often use this electrical analog, and the plant is the conduit. The transport happens from the soil to the air, plant is the conduit. And the analog is that the current is the water flow, the driver, in the case of an electrical circuit, a voltage difference. Here it's a water potential difference. And I'll explain what a water potential is. And all the elements in the plant are conductors with a resistance. And these are, the water transport happens through xylem vessels, which you see in the upper right, which are these conduits, these vessels, which are very tiny, but fairly efficient in transporting water, yet they have a resistance because they're so tiny. And there's also capacitors in terms of water storing cells and tissues, which I want to talk about now. The important thing to realize with water transport through plants is it's all driven by a suction from the leaves. And that's suction that is generated by the transpiration. We call it suction because it is actually a negative pressure. And we call it a negative water potential, which we will demonstrate how we measure it in the greenhouse. So that's the whole driver of the entire water movement through plants. It's actually a thermodynamic concept. It's a measure of the energy state of water, and the units it's measured in is in pressure units, pascals. So water flows from a high water potential to a low water potential, as would happen for voltages. So the water potential in the soil might be close to 0. In the leaf, it will be -1, -2, -3, -4. So how that develops, for example, in a drying soil, is an experiment shown here, a classical experiment by water relations pioneer. You see water potential plotted against time, and the solid line here indicates the soil water potential declining as the soil dries out. And the other solid, finer line with a diurnal pattern illustrate the leaf water potential, which comes back to soil water potential overnight and then declines to more negative water potentials during the day. Also plotted in the hatched line is the root water potential. So on the right to the diagram, we see this gradient shown as an example for the day 3. The soil water potential is -0.4, the root water potential -0.6, and the leaf water potential -0.2. So the more negative the leaf water potential is, the harder it pulls on that soil water to come out and be transported through the plant. Now we've established that plants contain a lot of water, but they use lots more water. And why would they use so much more water than they actually need is a simple reason: they need to have the stomata open. Stomata are the pores in leaves that allow CO2 to get in. And CO2, of course, needs to get in because plants need to photosynthesize to grow. So there's an unavoidable link between water loss and photosynthesis. And if there's not sufficient water, photosynthesis and growth will be impeded. Now, as I've said before, plants deal with lots of changing conditions, and they therefore need to regulate their flows. So if it's too hot and too dry in the atmosphere, they need to close their pores to some extent. So in response to drought, they will reduce their transpiration, and they may adjust their transport capacity through stems and so on. They may drop leaves, and they may grow more roots if given the time. But this is different for every species. So some species are better at certain things than others. And so they're different in how much, how, and how fast they will respond. So why is all this relevant to restoration? This gives us tools to evaluate stress intensity, to assess how tolerant or vulnerable plants are, and to diagnose what the cause of the stress is. And it can then ultimately help us to match species to the substrate and the climate. Now let's get to how we measure all of this. There's two pictures here, one from the field and one from the glasshouse. What we'll be demonstrating in glasshouse is all field suitable portable equipment, but there's other stuff that we're not showing here. So we can measure all of these things: the water flow, the water status, the potentials, and how they relate to soil climate, and can measure the implications for the health condition of the plants. But often we need to go back to the glasshouse to actually test things, because we need to be there to control the situations, the environment. We need to be there to do frequent measurements, and to do pretty closely controlled comparisons. And that allows us to understand the mechanisms, and allows us then to generalize our insights for certain plants to other plants. So this last bit will just be about a short introduction about the methods, aspects of the theory that you might not be able to understand from just seeing the equipment being demonstrated. So as I said, we are measuring suction in plants. These are negative water potentials. And the standard method to measure that is by applying a counterbalancing positive pressure. So we have a pressure vessel here that we can insert a plant in. We can increase the pressure in the vessel, and that will balance that negative pressure. On the right hand side, there's two images of the stem of the plant here, with the situation of an intact water column in that stem, in those vessels that we've seen before, going all the way through that piece of stem. Then when we cut it, because there's suction in the leaves, that water column will actually retract into the xylem of that stem, shown in the second image on the right. What we do by applying external pressure is pushing back that water column until it leaves that stem. And when we see that appearing on the surface of the stem, we know that we've just balanced that original negative pressure. And that's what Fiona will be demonstrating in the glasshouse. So they look like this. There's a pressure chamber, there's a pressure gauge, and there's a lid that can be adjusted to tighten that stem in place and be observed. Now for photosynthesis, we use equipment that's pretty advanced. We'll get the details in the glasshouse, but really the principle is about measuring the flow of gases in and out of leaves. And the gases of interest here are CO2 for photosynthesis and water for transpiration. The principle basically is that we have a closed chamber, not really closed, but it's not leaking apart from an inflow and an outflow. And we basically analyze the air composition of the inflowing and outflowing gas of that chamber. So for those of you technically inclined, we see here units of concentration given in moles per mole. So there's moles of CO2 per mole of air. Moles are numbers of atoms, basically. And that's what the device measures. So we have an inflowing air of 400 parts per million of CO2. That's the atmospheric concentration of CO2. And the outflowing air will have less because the leaf has taken up some CO2, because that's what it does when it's photosynthesizing. And in the opposite direction, for water, we see an increase in the concentration of water in that air because it's transpired. So from this, we get all sorts of information, the rates of transpiration, rates of photosynthesis, but also insights into physiology, into exactly how open those pores, those stomata are. And we can manipulate all the conditions that influence it, like light intensity, humidity, and temperature, to see what the effect on the results is. And we can decide how plants regulate these processes. and determine how species vary. The example underneath there is from a wheatland remnant common system where you see large difference between species in spring and summer. A related technique is this of chlorophyll fluorescence. You all know that chlorophyll is the pigment in leaves that captures the light, and it's that energy captured by the chlorophyll that's then converted into, through photosynthesis, into sugars. And the sugars are used as energy and building blocks, so for growth and maintenance of the plant itself. It turns out that plants always absorb more energy than what they can actually use. Particularly in stressful conditions, they have to get rid of a lot of that energy. So energy absorbed by the chlorophyll is not only used for conversion into sugars, but a lot of that has to be lost again as energy in the form of heat, or what is called chlorophyll fluorescence, which basically is chlorophylls having accepted that energy, then having to re-emit the energy into the atmosphere. Now, the re-emission of chlorophyll absorbs in the region of 400 to 700 nanometers, but the re-emission occurs in different wavelengths, in longer wavelengths with lower energy content. And we can measure those wavelengths. So we can measure how much energy the plant has to get rid of. And we know that plants in stressful conditions have to get rid of more energy, which allows us to take a measure of stress. Plants are unable to use that energy will have more fluorescence. So we can measure photosynthetic efficiency in a very straightforward, simple way. Here's an example from, again, a wheat belt situation, a mallee compared with she oak, where the mallee is deep-rooted, the she oak is shallow-rooted. The she oak has poor access to water in summer and has therefore very negative water potential, experiences stress, therefore cannot use all of that absorbed energy, and will therefore have high rates of chlorophyll fluorescence expressed as low rates of this ratio shown here. Sorry if I'm going a little bit fast. I realize I'm over time already, so I will skip this one. The last thing is really about, do we really need to use all of these complicated instruments? And one of our focus areas in this CMSR period has been looking for proxies, for simpler methods to use in the field to have similar sort of information as our complex physiological information. And one of them is spectral sensing. You all know that plants absorb radiation, but they also transmit some of that radiation, but importantly they reflect radiation, which we can measure easily. We can measure it when we stand next to the plant, but also by satellites from the sky or drones. So there's a lot of information in that because different wavelengths are reflected to different extent, and these reflections spectra correlate with leaf structure, leaf chemical composition, and physiology. So the next slide shows the spectrum from 400 to 2400 nanometers, and you see some indications there of chemical compounds that correlate with different bands in that spectrum that we can measure, and therefore get an index defined based on carefully chosen wavelengths that tell us something about the chlorophyll or the red pigments or the water content of tissues. And finally, thermal sensing is also quite easy to do from a distance, as I've mentioned. And interestingly, foliage temperatures are very strongly influenced by transpiration. And you're all familiar with the process of evaporative cooling. And that happens when leaves transpire, they get cooler than the atmosphere. So the cooler a leaf is, the more transpires basically, and that's shown in the graph on the right. So if we simultaneously measure the leaf and the air temperature, we can estimate transpiration rates. And this next image shows a thermal image from a thermal camera that we'll show downstairs in the glass house, which is non-contact infrared sensed temperature sensing. And there's 3 leaves in here, which are called purple, blue, and green, and they're also in the graph. The green leaf is not transpiring, and we've ensured that it's not transpiring by putting some paraffin oil on it, and it therefore cannot get rid of that water, is not cooling. The purple leaf we've wetted by spraying water on it, so that's fully losing water at a very high rate. So that is the one that has the lowest temperature here in the top graph, and the dry leaf has a very high temperature, and the naturally transpiring leaf is the blue one, which according to how much it transpires will be more to the left hand side, therefore hotter, or more to the right hand side, therefore cooler. So how well does this all work? So I don't want to take away too much of Jaume's messages. This is what his thesis is about, and we're finding that there are significant correlations between these thermal and spectral indices and the physiology, but they're not always accurate for all the species, and often they're species specific. With temperature, we're finding that they're very variable, and we know indeed that transpiration is only one of the factors influencing leaf temperatures. One of the issues that we have in restoration environments is there's so much background, so much soil and litter, which has very different signals from the leaves. So when plants are small, we have difficulty, but they may be overcome with high resolution systems. We have shown great promise for clip-on sensors, and we'll demonstrate some of them in the glasshouse. All right, so time has come to conclude this session, and I will actually run down now as quickly as possible to not waste more time and take the full opportunity to get people in the glasshouse to start doing demonstrations. I will see you there. The last picture is from a drone image, a 3D thermal image, where you can see the cooler canopies of the Banksia canopy in the Banksia woodland. My email is on screen for any questions that you don't have a chance to get answers to in this session or at any time later after today. Please contact me. I will see you in the glasshouse shortly. And I now have to find a way to unshare. Yes. Over to Fiamma, who's leading the crew in the glasshouse.
Hello everybody, quick set change over to the plant growth facilities at the at UWA. So here we are in one of our glass houses. It's one of our hot glass houses, so apology if we all look a little bit sweaty, but let's have a look inside while Eric makes his way down. The plant growth facilities here at UWA are set up to run all sorts of experiments under irrigation, under different temperature conditions, and to be able to use with various pieces of equipment, not just ecophysiological equipment. This glasshouse, as you can see, has several experiments running in it at the moment, including one from the ecophysiology node. For those of you who are familiar, it is our double drought experiment, but essentially it is an experiment where we're hoping to see the effects of drought conditioning not only on plants but on soil microbes. I'll take you over to have a look. Here it is. So you get to see an example of an experiment in process here in our glasshouse facilities. So it is using 3 pilbara species, an acacia, a trodia, and a eucalypt under drought and non-drought conditions. This is the second phase of it. Hopefully some exciting results coming your way soon. Here is Jama setting up for one of our experiments, one of our PhD students you'll be hearing from more shortly. The wonderful Wei-san, who's going to wave at you right now, is being our brilliant camera woman. And for those of you who I haven't met, I'm Fiamma. Let's make our way over to the pressure chamber. Okay, so as Eric explained to you, one of the pieces of equipment that we use in ecophysiology, one of the very sort of traditional pieces of equipment is the pressure chamber. And we use the pressure chamber to measure leaf water potential, or the amount of pressure which is required to release xylem fluid out of the xylem. So essentially it's how tightly the plant is holding on to its water, and essentially the more pressure that we need to apply, the more tightly the plant is holding on to the water, and therefore we can infer something about its stress or its ability to cope with stress. So I'll get Wei-san to come in and have a little zoom so you can see all the components of it. It's more colloquially known as a pressure bomb or a pressure chamber. So if we follow the pipeline down there, you can see that it's connected to gas. The gas comes up the pipeline and into the chamber here. And here is the gauge that we read the pressure chamber with. I'm actually going to do a demonstration. We were hoping for cloudier conditions today and better light, but hopefully we'll be able to show you how it actually works in real life. So here are some plants under various stages of stress that we've set up for you. I'm going to take a eucalypt leaf and demonstrate the leaf water potential for you. Eric has arrived. I might get him to take over commentary just while I concentrate, since we'll be under high gas pressure, if that's okay. Okay, so for leaf water potential, we take usually a leaf. Sometimes it has to be a small stem depending on the type of leaf that we have. But it fits in this head so that the petiole of the leaf is visible from the top, while the bulk of the sample of the leaf is in the pressure chamber where we can apply the gas. So I'll get Wei-san maybe to have a little zoom in. See if you can see the petiole sticking out there. Excellent. Okay, I'm just going to get myself set up. Excellent. Okay, so I'm just going to turn the gas on now. So the gas is going to start applying pressure to that leaf inside the chamber. And once there's enough pressure, we're going to get water or the xylem fluid starting coming out of that cut that I've made. And the amount of pressure that is required will be our leaf water potential. Okay, so as you can see, this requires a little bit of time for each sample, and the more stressed a plant is, the more pressure we need to put in and the more time it takes. So excuse me if this takes just a moment, And hopefully we'll be able to show you this as the liquid comes out. Okay, so I'm going to take a reading now at negative 1.3 megapascal. So that is actually a fairly happy plant in terms of water. And I'm going to step back now, and hopefully Wei-san will be able to show you the bubbles coming out of that cut end. Thank you very much, Wei-san. Okay, so this is one of those sort of key fundamental core pieces of equipment, excuse me, it's warm in here, in ecophysiology. I've used it a lot in the field. I love the fact it's indestructible and it's analog, right? You don't need a digital scale, batteries don't run out, etc. But as you can see, it is a little bit time intensive. If you imagine you had to do many leaves per plot per day, that it is a little bit more time intensive. So pros and cons. Eric?
Yeah.
Anything else? You certainly can.
Yeah, certainly very robust technique and has been around for decades. What hasn't been around for so long is the measurement of photosynthesis. You used to need full truckload of equipment for that. And it's certainly not as robust either. But it is quite usable, and we use it a lot in the field. So if Wei-san can close up a little bit here. So this is what we in short call the LICOR, a gas exchange system, which basically is a console and a leaf chamber. So when we close up here, we have a space here where we can insert a leaf, and that is where the inflow and outflow of is measured. So all the measurements are done either locally here in the chamber, or through being sensed and being recorded in the console. So what the console does, it prepares this airstream, there's a pump in there, flows here continuously, and the analysis is actually done here, and that analyzes for CO2 and water. So that is basically the principle, but the beauty of the instrument as well is that we can set all sorts of things. We can control the temperature here. We measure it, of course, all the time. We can use, as we have here, use an artificial light source. And you can see it's an artificial light source because the color, these are LEDs that are very efficient in providing light. And we can manipulate the composition of the air as well to change the CO2, for example. Is the sound quality good? Do we know that people are hearing us, or might be worth checking? Yeah. Okay. So yeah, taking a leaf and putting it inside the chamber just like that, and from that point it is being measured. Now we'll do that in a minute, just to show you different types of results. But I want to show you first what the data come out of. So can we switch to this screen, then? Or are we gonna? Okay. Yeah. All right. Is that well positioned there? So I will now do a measurement. Oops. Okay, that's okay. I'm just trying to see how we can easily do that. I'll first show the screen there, just to introduce to you what screen looks like, and you just basically see lots of numbers there. On the top row, we see the CO2 concentrations, and you see at the top left, right, you see maybe, if the light is okay, that the CO2 concentration is pretty much 400, which is around ambient concentration. Now the next one, you see another value that's also 400. That's because there's nothing in this chamber now, so the inflowing air and the outflowing air are identical. And you see that same happening for the water on the top right. So there's lots of measurements here until the machine calculates first a rate of photosynthesis, the opening of the stomata, the transpiration here. I won't go into detail about what the rest of it says. There's a lot of settings, a lot of manipulations, a lot of different ways of viewing data, which you don't need to know right now. I'll just want to demonstrate you what happens when we start measuring a leaf. So if my son just stays on screen, I will now quickly open the chamber. And you should very quickly see this sample CO2 going up, because I was there breathing CO2 from my breath, and you see how fast this machine responds. Now we can also see these fast peaks when we plot these data. So we've plotted here photosynthesis on the left, and the stomatal conductance, the opening of the stomata on the right-hand side. And when I was blowing air into that chamber, you can see this massive peak, which then soon goes back to that same stable 0 value that it was at before. So we'll now do the same, but instead of blowing into it, I will test a leaf, and I will first measure this Heikia. You can show it quickly. This is a Heikia prostrata from around here. It's been well watered, so it should have a reasonable activity. And I'll introduce it in the chamber. And we can go back to screen. And we'll have a short transition period where which is again due to the chamber being open. The scale of the screen self-adjusts to the maximum and minimum values shown, so we won't see the details because we have this massive peak here. But that will go off the screen really quickly. So in the meantime, we can already look at the values here. For photosynthesis, we see a value of 11, and it's still sort of stabilizing a little bit, but it's around 11.3, and the conductance is 0.38. So this is, from our experience, a very healthy leaf, photosynthesizing pretty well. And you see the response after we introduce the leaf, it's now leveling down. So we're pretty much ready to take this as a measurement. And of course, this console has a memory where we just click a button and it will be stored in memory. So just to remember, for our next measurements, we're sitting at a photosynthesis of slightly more than 11 and a conductance of close to 0.4. So I will now change to another plant, which is this one, which has not had a lot of water lately. So we'll change over. Take this one out quickly. introduce this one. So again, we will have a short transitional period. And apart from looking at conductance and photosynthesis, we can also have transpiration if we want to. We can look at the actual CO2 values that you see here, or the water values that you see here, the temperatures. This is the air temperature here, and this is the leaf temperature. You see that the leaf temperature responded quite strongly when we actually introduced the plant in it. And I'll just move back to photosynthesis. It's starting to stabilize, but we still need to wait until this disappears off the screen. But we see the photosynthesis stabilizing at about 3.5 instead of 11, and the conductance at 0.04 instead of 0.4. So there's a large difference between these two plants of the same species according to whether it's been watered or not, exactly as expected. Now, if we have time, we will measure other things on these leaves as well that might be correlated with photosynthesis. So that is, oops, okay, it's gone. That is what we can do with this machine. I think for the sake of time, we probably better move on. So when I spoke from my office, I mentioned chlorophyll fluorescence as a way to look into photosynthetic efficiency as well. So we have this equipment here. This is the very small handheld, very handy field equipment, which Fiamma will demonstrate on the same plants. No, not on the same plants. In this case, we will go for the eucalypts. What you'll see here is they have clips on them. They go with the handheld equipment. The reason that they have clips is that this measurement is best done on plants that have not had any photosynthesis for a while. So the photostatic activity has gone down to 0. And what the machine will do then is will expose that leaf to a light flash. And because it hasn't had any photostatic activity in the past half hour, it will fully demonstrate its capacity to photosynthesize. And if so it will then give a single index value of the fluorescence, which will tell us how healthy that leaf basically is. So we have two plants here of the same eucalypt species. One of them is quite senescent, has all these red and yellow pigments, probably not so healthy. This one is a lot greener. So we'll just do a few measurements on their leaves. Over to Fiamma.
Wait. Great. Okay, Wei-san. So I might get you to have a quick zoom into the clips so that people can see them clipped on there. Yeah. And so the difference between those two leaves, these two clips here, are that this one has its little window, it's open, you can see, and then we slide the little window closed. So as Eric said, the idea is that the piece of leaf that would appear in that little window has had no light. So we put these on about an hour ago, but in the field it's obviously not that long. So I then place, Wei-san can see there. Yep, so I then place the machine into the slot that the clip provides. I slide back and open the window. And we can see that we've got a reading there of 0.77. So in the grand scheme of things, that's actually not too much of a stress plant. we're going to take one on the healthier plant. Again, I'm just going to slot that in, slide the clip back. It's 0.82. So that
A little bit higher.
A little bit higher. And we'll do one more. I might do this one here, it's a little bit easier to get to.
So despite it looking pretty bad, it's not actually doing so badly. So this leaf was actually exposed to some chilling early in the morning, and it hasn't suffered at all from it.
0.82 as well.
Excellent.
Yeah.
So they're very, very resilient plants. When you get very stressed plants, the values that you'll get from this will go as low as 0.3. So in actual field work at mine sites, we've had the whole range of values from the 0.8, which is roughly what you expect for healthy plants, to down to 0.3. And it's a very accurate measurement of the photostatic efficiency. And to some extent, it's something that can be measured remotely as well. We might get to that with Dauman in a minute. What is our next thing?
You want to show the barometer.
I think, what is the time? We might need to skip the barometer. Yeah, I think we'll skip the barometer and move on to our attempt to work with alternative sensors, because the reality is that some of these sensors, the fluorescence is not difficult, but the gas exchange and the water potential are logistically sometimes hard to interpret as well, to troubleshoot in the field, and sensitive to harsh conditions as well. And we don't have very many of those instruments that we can do at the same time. So if there's quick and easy measurements that are reliable enough to give a proxy for these standard physiological measures, then that would be really advantageous. And that's one of Jaume's main objectives here. Over to him.
Thank you, Eric. Yeah, so as Eric was saying, the idea behind exploring remote sensing as a method to assess plant physiology is to find quicker and more efficient ways of doing it. Now, the methods you just saw being demonstrated are very precise, but they're sometimes difficult to perform. So in an area such as a restored mine site, where we want to have a good idea of what's happening over a very large area with a lot of plants, we want to know if there are methods that can help us do that more quickly and more efficiently. And that's why we've been researching remote sensing as a potential indicator. Now, you all might have heard about remote sensing, and as a concept, there are many different levels at which remote sensing can be done. There are satellites that sense light from the top of the atmosphere or from space. There are drones that can have sensors mounted on them. And then there are also handheld sensors that we can use from the ground. And those last ones are the ones that we have used in this project, basically because they allow us to precisely establish the relationships between the proxy indicators that we get from remote sensing instruments and the actual variables that we can measure with instruments such as the ones you just saw being demonstrated now. So today I'm going to be demonstrating the use of two of these sensors that we've been using. The first one is a field spectrometer, which is this instrument you can see here on the table. What this does, it measures reflected light. So it measures how plants interact with light and what amount in each part of the spectrum is reflected by the plant. And we can use this information to retrieve physiological status of the plant, basically because it's telling us about physical and chemical properties of the plant. We can infer some of the attributes that we could assume the plant to have in terms of its physiology. So with no further delay, I will demonstrate a bit how to use this spectrometer. Let me just move the trolley next to the table. So here we have this plant, and today I'll be showing you how to use this leaf clip, which basically allows us to take very precise information from leaf reflectance. So the spectrometer has an internal light source that you have to turn on. It has to be calibrated against this white surface, which I've already done. And the reason we have to do that is because this white surface reflects everything, all the light that comes on it. And what we're going to get from the leaf is a proportion of that reflected light. So what proportion of light in each wavelength is being reflected by the leaf? So I'll get a bit closer. Not sure, Wei-san, if you can come here and show maybe first how to clip the leaf. So we put on the leaf here, we clip it in, and we take a measure. And that curve you can see there on the screen is the reflectance of this particular leaf. You can see the leaf is reflecting light differently in different parts of the spectrum, which is what you can see on the x-axis. And what I've done is basically I prepared before this demonstration 2 leaves that look very different. And as you can see here, this is the actual spectrum of 2 leaves of a healthy plant and of a stressed plant. And what we do with this data is we basically analyze it in many different ways, and we see how we can correlate it with the measures that you just saw being demonstrated, being leaf water content, fluorescence, or we can also measure or infer pigment content using this instrument. And pigment content is actually, will take a few days for us to be able to get pigment content of a plant. So the fact that we're able to retrieve it using this instrument is very helpful in terms of monitoring large areas such as restored mine sites. Now there's another instrument that I wanted to share with you today, which is this, it's a polypen. So basically the polypen is a small version of the spectrometer. It can measure a narrower part of the spectrum, but it's much easier to carry around. The spectrometer can be carried around. You can mount it on a backpack. The laptop goes on a tray, so you can walk around an area and take measures of many plants. And with the polypen, again, what we need to do first is we need to calibrate it using this, this standard reflectance surface. We need to mount it here. You can see here. We mount it on the leaf clip area. We place on calibrate. Calibration in progress. Calibration done. We press OK. And now we can go onto our leaf of interest and clip it in. And then get reflectance. It already has it. It's very quick. And so what we can see here is a similar curve to what you saw on the laptop, but in a narrow part of the spectrum. And you can also see it's giving us a few spectral indices down here, such as NDVI and another one here. So we can actually program these indices for the polypen to give us the data that we're interested in. So this is in terms of reflectance. And there's another method that Eric mentioned earlier, which is thermal imaging and measuring plant temperature. Now, I will probably grab the other plant because it's a bit more suited to these measures. So here we have 2 different, what we call model leaves, right? So one of these leaves has been covered in petroleum jelly, which means that we're simulating a very stressed leaf that is not transpiring. And the other one, we're gonna spray it with water. So we're gonna simulating that is transpiring at maximum intensity. And what we can we can do now with the thermal camera is we can basically, we'll be able to see the difference very clearly. If you, I'm not sure if you can come here with Sun and focus on the screen. All right, so let me focus on that. So you can see, so this is the sample leaf, right? You can see, and this is the wet leaf, and this is the dry leaf. So you can see there are 3 different colors, which means there are in 3 very different temperatures. And the sample leaf is somehow in between the dry leaf and the cool leaf. Now, what we can do is when we take a thermal image of this, for example, I just took an image and I stored it. What we can do later on is we can analyze this data in the computer and basically try to estimate how much the plant is transpiring using a series of quite complicated energy balance equations. And once we've done that, we've realized that we're able to estimate trends, but we have problems getting very similar measures to what we would measure with, for example, with the LICO, because it's very hard to approximate these physical parameters that affect the leaf's object. But it's still valuable information. And lastly, what I wanted to share with you, and this is some data from the field. Basically, I have a few plots here, which is one of the plants growing in Hansen, restored mine site. And you can see here we have the measured leaf water content against the predicted water content. And we're getting a pretty good relationship there. Here we have chlorophyll content, which I was telling you earlier is quite complex to measure in the lab, takes a few days. We're getting a pretty good correlation as well using data from the spectrometer. And we also have fluorescence, in which relationship is not so clear, but there's definitely a trend and some valuable information. And finally, stomatal conductance. We have here measured conductance against predicted conductance. You can see that we're overestimating conductance, which has to do with the physical parameters we use in our equations, but we still get a pretty good trend. And what we have seen is that when we combine all these data together in a multivariate statistical analysis, we can actually track seasonal change of different species in the field in a very similar way that we can do using traditionally measured physiological variables. So there's definitely potential for us to explore how to use remote distance data in order to track seasonal change in plants and to see whether some plants are maybe going into trouble in a time of the year where they shouldn't. And that's all from me.
Great. Thank you, John.
No worries.
Can I have your microphone?
Yep.
So I think that shows very clearly that not only can we measure the real physiology that scientists like to measure, but also practical measures in the field, quick measures that are meaningful, correlate well, and allow you to diagnose or understand plants and how well they perform. I just wanted to add to the thermal one that it can be even more practical than having this thermal camera and having to process those data in the field. In your office, you've all seen the temperature guns that doctors and airport personnel use. Of course, you can use that as well for your plants, as long as you measure a reference temperature with equipment like this, which costs a couple of $100. That will all give you relative measures, but sometimes that's the only thing you're after, to see if one species does better than the other, or if a species in one location does better than in another location. So I hope that we've given you a broad idea of what sort of things we can do. We haven't shown you every tool in our toolbox. This is how we work, how we learn, and how we hope to convey information to you as practitioners in the field. And as I think we've shown, there's some of these things that can be applicable by yourself as well. So I think we're getting close to our end time, and I want to give people a chance to ask some questions. Have you looked at the chat at all? Okay. Open for questions then, please. Our research is ongoing. We're working very hard to get everything done by next month, basically. Theses are being written, papers are being written, reports are being completed, and you'll see all of that become available very soon. No further questions? Okay. Any comments? Of course, if people want to ask questions later on through email, very welcome. If you're interested in resolving things that you're coping with at your site, also very welcome to have a chat, which can lead to a few exploratory measurements or whole projects. We're flexible. All right. So if there's no further questions now, yeah, do feel free to contact us along the email way or by phone. And thank you very much for attending this session. I hope you found it useful and have given us a good impression of what ecophysiology is all about. So we'll leave it there. I think thanks to all those who helped here and in the office, and those who couldn't make it today. The session will be recorded, I think, so it should be available for those who couldn't make it. Thank you and see you soon.
Thank you, Eric.